In this video, you will learn about the neuromuscular junction and the mechanism of excitation-contraction coupling. The neuromuscular junction is the point of communication between a motor neuron and a skeletal muscle fiber. It consists of three main structures. The axon terminal, which is the end of the motor neuron. This terminal releases the neurotransmitter acetylcholine to excite the muscle fiber. The motor end plate, which is the specialized region of the sarcolemma. It contains nicotinic acetylcholine receptors, which detect and respond to the neurotransmitter released by the neuron. The synaptic cleft, which is the tiny space between the axon terminal and the motor end plate. Neurotransmitters must diffuse across this gap to stimulate the muscle cell. Now let's connect this to excitation-contraction coupling, which is the process of linking the electrical signal from the neuron to the actual contraction of the muscle fiber. Here's how it works step by step. A nerve impulse reaches the axon terminal of the motor neuron. Voltage-gated calcium channels in the neuron's membrane open, allowing calcium ions to enter the terminal. This influx of calcium triggers the release of acetylcholine into the synaptic cleft by exocytosis. Acetylcholine diffuses across the cleft and binds to receptors on the motor end plate. These receptors are ligand-gated ion channels. When activated, the channels allow sodium ions to enter the muscle fiber, causing depolarization. This initiates an action potential in the sarcolemma. The action potential rapidly spreads across the sarcolemma and travels down into the T-tubules. This electrical signal triggers calcium release from the terminal cisternae of the sarcoplasmic reticulum. Calcium then binds to troponin, which shifts tropomyosin away from the myosin binding sites on actin. With binding sites exposed, myosin heads attach to actin, and contraction begins. One action potential releases enough calcium to stimulate a short burst of contraction called a twitch. To end contraction, calcium is pumped back into the sarcoplasmic reticulum. Without calcium binding to troponin, tropomyosin will move back to cover the myosin-binding sites of actin, allowing the muscle to relax. In short, the neuromuscular junction initiates the signal, and excitation-contraction coupling carries that signal into the muscle fiber to activate the myofilaments. This precise sequence ensures that every nerve impulse produces a coordinated muscle contraction.